All right. I know more people likely will be trickling in, but just to kind of make sure we are on time, welcome everyone. I'm Dr. Lauren Schmidt. I'm the Chief Science Officer at the Phelan-McDermid Syndrome Foundation, and we are so happy to be hosting this Gene Therapy 101 today. Thank you for the American Society of Gene and Cell Therapy for co-hosting with us and making this happen. And we're especially grateful for Dr. Kim Goodspeed, who will be presenting today. She is a child neurologist and the medical director at Ultragenyx. So thank you again for tuning in. And please put any of your questions in the Q&A, and we will make sure that Amanda and I will be asking those at the very end. Enjoy. I'll hand it over to you, Kim.
Actually going to go to Allie first, then me.
Perfect.
Hi, everyone. We're excited to be here. I'm the Senior Patient Outreach Manager at ASGCT. So I just wanted to introduce who we are. We are a professional membership organization for researchers, clinicians, and advocates working in the field of cell and gene therapy. So we want to advance knowledge and awareness of these therapies and increase the access to treatments. So we work with members such as Kim, who are working in the field and doing this research and working with different groups and patients to move these therapies along. And we do a ton of different things. Patient outreach is one of those. We also do global outreach. We have a big annual meeting, scientific journals, lots of other events. But we saw a big need to reach out directly to the patient community to make sure there's an understanding of what cell and gene therapy is. As we know, it can be really complex. And it's quite a long journey for a lot of groups to be a part of. So we're excited to just connect with your community today to hopefully bridge that gap and help you understand this a little bit better. You can go to the next slide. One thing I wanted to make sure you all are aware of is we do have a patient education website. There's a lot of patient-friendly resources on cell and gene therapies, clinical trials, and things just to help explain how this works. There's short animated videos, some infographics. So if you want to go ahead, you can use the QR code to check out some of those resources. Next slide. And then also 2 other ways you can interact with us. We have a newsletter that comes out every other month. This is the easiest way to subscribe and just stay up to date on any other events we have going on for the patient community, different resources we think would be relevant or news in the rare disease space with other things going on in the pipeline. So you're welcome to subscribe to that and follow along. And then also in March, we host an Empowering Patient Summit, and that's a free virtual event you can attend just to hear the latest education and areas of need on what we need to discuss in this space. So we would welcome you to register and join us for that as well. And you can stay tuned for that in the newsletter as well. So next, I'm excited to introduce you to Dr. Kim Goodspeed. She'll be doing the majority of this presentation. She's a child neurologist and neurodevelopmental disability specialist. She is currently a medical director with Ultragenyx, focusing on genetic therapy development for neurological disorders. And previously, she was an assistant professor in the UT Southwestern Department of Pediatrics in neurology. There, she conducted clinical and translational research in rare genetic and neurodevelopmental disorders. So I will hand it over to Kim. Thank you.
Thanks, Allie. And I'm really excited to be back with the PMS community. One of the first projects I got to work on at UT Southwestern as a resident actually was in PMS. So I've been in and around your community for a while, and I was really excited to get the opportunity to come back in this capacity. We are going to go through a lot of stuff today. So I broken into kind of goals, and then we'll hit 3 main objectives, and it's going to feel like a little bit of a whirlwind, I'm afraid. But the goal is that we demystify this. We make it feel a little more accessible so that the next time you hear this information, you're like, oh yeah, I've heard that once before. And every time you hear it after that, it gets a little more comfortable each time. And then that knowledge can then empower you to help make good decisions for you and your family, or in discussions with your healthcare providers. If you are a healthcare provider, maybe you're new to this field and you're picking up stuff to help empower your patients to make good decisions that make sense for them. So we're going to do a quick overview of genetic therapies, and then we're going to kind of run through some nuts and bolts of clinical trials and what it means to be a participant. This photo, I'm a Texan, so this photo is actually from Canyon Lake. It's probably not quite this full anymore because it's been in a massive drought lately, but this is a really beautiful dam where I think it's a nice picture of like we're heading somewhere, but that somewhere is always continuously moving, right? This field is continuously evolving and updating, but we're always heading in the direction of new and improved. And so I love this image that my husband sneakily took from behind me. But I think it's a really nice way to think about like there's this big body of water. We're kind of containing it and organizing it, but we're also moving everything forward. And so this presentation may be out of date in a couple of years. And that's how quickly this field is really moving. So we're going to set it up by going through what genetic material is and then hitting some of the high points of these different genetic therapies without going into too much detail on any single one of them, but at least gives you some degree of familiarity with them. So when we say genetic material, what does that mean? So in our bodies, it kind of comes in two very basic forms. It gets a lot more complicated very quickly, but there's essentially DNA and RNA. So DNA is basically where we are storing our genetic information. So I always used to use the analogy of encyclopedias. So you get a set of encyclopedias from mom and a set of encyclopedias from dad, and that makes up your genetic code. Some of those pages of the encyclopedias have really critical information on how to make proteins. That's our genes. And then other parts of the DNA have kind of blank or intervening pages, or maybe pages like a table of contents that directs you where to go find information. And our DNA is like that too. So it's got parts of it that make up proteins and parts of it that are used just for guidance and regulatory and making sure that all the different proteins get turned on at the right time. But that's the storage compartment. RNA is really that more functional compartment, the more functional version of genetic material, where it makes a copy from the DNA that can then be used in a lot of different ways. So when we think about DNA to DNA, that's replication. You're just making a direct copy, staying in the same type of material. But when we go from DNA to RNA, that's actually called transcription. And when you go from RNA to a protein, that's called translation. There's no way to go directly from DNA to a protein. So you have to have this intervening step of RNA. And all of this lives within the cell. So we've got the nucleus that's like the central command place for the cell. That's where the DNA lives. All the instructions and critical material are housed and stored there. RNA can kind of come in between the nucleus as well as the surrounding parts of the cell, or the cytoplasm, or kind of the community portion of the cell. And so RNA gets copied from DNA. It can go out into the cytoplasm of the cell, and that's where it can make a protein. And all of these proteins are critically important for different functions in our body. So then when we think about therapies, we can kind of break it down similarly. There's DNA therapies, there's RNA therapies, and then there's this other thing called an ASO, which is an antisense oligonucleotide, which is kind of a synthetic hybrid of the two. So in DNA therapies, typically these are the ones that are just given once. You're directly giving somebody back a gene or a copy of a gene, or in y'all's case, because SHANK3 is such a big gene, they've made a mini gene or a smaller version of that has just the critical elements in it. But you're giving that gene back to the patient. And the goal is that that DNA gets delivered into the nucleus and it can be copied and used and turned into RNA so that it can make a protein. But it's essentially like a supplemental copy to your encyclopedia because there was something wrong with like the SHANK3 gene for y'all. In RNA therapies, you're kind of taking that middleman approach. So you're engineering some sort of an RNA. There's a lot of different ones of these. And this is probably one of the fastest growing fields in gene therapy right now, that can change how different proteins are made, say upregulate, maybe silence them, but it's an RNA molecule instead of a DNA molecule. DNA is a lot more stable than RNA. And so for most of our RNA therapies, you're going to need to get multiple treatments over time, as opposed to a DNA treatment. Typically, we're just giving that one time by current today standards. And then, as I said, ASOs are kind of that in-between-Z place. So it's a synthetic molecule. It's kind of manufactured in a lab, and it may have some components of DNA, some components of RNA, and they can get kind of fancy with how they do the chemical makeup and structure of it, depending on what they need the molecule to do. But these can go and kind of regulate how genes are expressed. So they're not direct copies or instructions on how to make a new protein. They're more controlling which proteins get turned on and off. So they're very commonly used to silence, so you knock down expression of a protein, but in some cases they're also used to upregulate or increase production of a certain protein. But think of them more as like a regulatory element as opposed to the direct instructions on how to make that protein itself. These also need to be given over a period of time. So you need repeated doses of both ASOs and typically of RNA therapies as well in order to maintain that therapeutic effect, whereas a one-time dose of a DNA therapy could potentially be permanent. And then we can think about, well, okay, so those are the different types of modalities that we have, but what can they do? And so that's when you can think about, what is our goal? Do we have too much of something? Do we have too little of something? And we need it to be Goldilocks and be just right. So, oops, excuse me. So if we've got gene addition, that's where maybe you're missing a copy of a gene, or the copy that is in the individual patient isn't working. And so we need to just give them that supplementary copy back. That's what we can think of as gene addition, or gene transfer therapy. A lot of times right now we're using AAV, especially AAV9 if it's in a neurologic condition, but different types of viruses. The AAV part of that is the virus that's carrying the gene that can be changed and is updated over time. Gene silencing is another option. So let's say you've got an overactive gene, or you've got too much of a protein being made and we need to silence it or decrease that amount of protein. In my case, I'm managing our Angelman program, and we have an antisense oligonucleotide that knocks down a regulatory element that is silencing a gene. So we're silencing a silencer with an ASO. So there's a lot of interesting ways that we can start to apply these different techniques or approaches to alter and get more typical expression of genes and proteins depending on what's going on in that individual patient. Gene editing is another one that's kind of interesting and a newer modality or a newer approach. It's been used for a long time in making animal models of diseases, but now we've seen our first gene editing approach, or CRISPR technology, be approved for sickle cell. So we are seeing this come into the clinic, and it's being applied more and more. And this is where you have a little piece of DNA essentially that can go in and edit or correct a mistake or a mutation that exists in a gene. This is really helpful if you know that you've got a population where there's a particular mutation that's really commonly seen, and you can target that specific mutation. And then kind of out by itself sometimes, I feel like, is our modified cell therapies. These are often used in the cancer space or the oncology space, and CAR T cell therapy is one example of that. And essentially, you take cells from a patient, you genetically modify them in the lab to maybe engineer that they now carry a working copy of a gene, and then you give those cells back to a patient. And in the case of some oncology disorders or diseases, they can actually engineer them to kill the cancer cells. So the other three are mostly kind of therapies that are delivered directly to a patient, whereas this modified cell therapy is usually taken from the patient and then given back in kind of a two-step process. So explaining that a little bit in a little different way, in vivo treatments are what we call, when we, this top portion of the graph. So we have a gene that we want to package into a virus, or this is kind of depicting a lipid nanoparticle, or some sort of genetic material. We're going to package it in some sort of way, and we're delivering that directly to the patient. As compared to ex vivo, where cells get removed from the patient in step one, They get engineered in the lab to have the correct genetic material in them, and then those get delivered back to the patient in that kind of two-step process, excuse me. So in vivo, you go from the lab to the patient or the manufacturing facility directly into the patient. And then with an ex vivo approach, we take cells from the patient, engineer them, and get them back into the patient. There's pros and cons to all of this, and it really depends on a lot of different things, the size of the gene, the patients that we're treating, the different symptoms those patients have. So there's a number of different factors that go into the decision making of which avenue you're going to take. And then we have to also think about, well, we're going to give it to the patient, but how are we going to give it to the patient? And so there's a number of different places that we can administer gene therapies, depending on what we're trying to do with that therapy. And so here's just few questions that scientists are asking themselves as they're designing these and developing these new treatments. Are we going to be giving it in vivo or ex vivo, like we just talked about? Which organs do we need to target? If we need to get to the liver or the blood, do we go directly into the blood versus do we need to get to the brain? We want to get something that's closer to that target organ. What dose are we going to be used? What did we do in the preclinical studies? Do we need to mirror that? But really and truly, I'm a neurologist at my core, and I know I'm speaking to folks that are affected by neurodevelopmental disorders. And almost always we're talking about delivering directly into the brain. A lot of times we need to get in there with a bigger dose than we're able to use if we go through the IV, because you have to give a really big dose in order to get it into the brain if you're going through the IV or intravenous route. If we go directly into the spinal fluid, in any number of these different ways, then you can usually use a slightly smaller dose and still get a big impact potentially. So a lot of times you're going to be hearing things like intracerebroventricular or ICV, or intraparenchymal, which means directly into the tissue. ICV means into the water sacs or the ventricles that hold the spinal fluid kind of in the center of the brain. And then the least invasive or less invasive than those two are going into the spinal fluid around the spinal cord, either in the lumbar lower back region or in the cisterna magna, which is kind of the lower part of the skull, top of the spinal cord region. There's a lot of different avenues. There's even some devices that are out now that are trying to improve our ability to deliver drug to the brain. But these are some of the common terms you may hear as different therapies are coming up the pike for you guys. So that was gene therapy in like 5 minutes. So by no means meant to be exhaustive, but hopefully gives you a little bit of an introduction and some direction and offshoots to go to kind of compartmentalize and organize this information. So the rest of the talk, we're going to talk more about clinical trials, because I know that's something that is on the horizon for you as a community, and it's big decisions. And again, it's a whole new vernacular, whole new vocabulary to be thinking about. So the first part, I'm going to think more about how they're designed and then what it's like to participate in them. So it's a long process from the bench to the bedside. So from preclinical studies, we're almost always starting with these studies in animals or cell culture, so that we're going into studies in non-humans before we go into humans. We need to understand the toxicology, what are the potential side effects, what are the potential doses, all of those good things. And then we take that information plus some of our initial clinical information in our plans, and we file an IND, or an investigational new drug application. That gets filed in the US to the FDA. And then of course there's other FDA equivalents across all the different global regions. And the next step is to do a phase 1/2 clinical trial. We'll talk about what that means exactly, but it's an initial study in the patients who have this condition. It's usually a smaller study, typically open label, meaning everybody's getting drug and everybody knows it. And you're looking primarily at safety, but also we'll look at some efficacy at endpoints as well. So is it safe and does it work? Once you have this data in hand, you go back to your FDA or equivalent health regulator in your region and share the data with them. And what are your plans to really confirm the findings that you had in your phase 1/2 study? So we think it's safe and effective, but we need to do another study, which is a phase 3 study or a registration enabling trial. And this is the study that is more rigorous, typically bigger, often has a control group where you're comparing treatment versus no treatment in some sort of way. And this is the study that will give us data that helps the regulator decide to approve or not approve. Once it's an approved therapy, any doctor, well, mostly any doctor could prescribe it. For gene therapy, sometimes we're talking about more specialty centers, so it may be a difficulty of, let's say maybe a rural community hospital that isn't as well resourced with the equipment necessary may not be able to administer, but it's essentially approved for use for a doctor to prescribe. And then we think about our phase 4 trials, and that's really just that kind of long-term efficacy, long-term safety. What are the things we didn't know because we just didn't have enough time to know them yet? And so we continue to follow patients even after drugs are approved. And the highlight I want for y'all is to take away is that your voice matters. And so there are ways to interact with regulators or with sponsors of clinical trials to let them know what you care about as a community, what symptoms matter to the community, what things would be impactful for you, what you would consider a success. And so all of this information is really important. And so staying connected in your case with PMSF or whatever patient advocacy group you're working with, is really important to make sure that you are staying engaged and offering that feedback when it's asked. And so taking each of those overview steps into a little bit more detail, on the preclinical side, there's a lot of stuff happening, typically all at once. So researchers like I were working with patients and trying to do some phenotyping. What do their EEGs look like? What do their brain MRIs look like? What do they look like clinically? What symptoms do they have? What do their exams look like? And then my colleagues in the basic sciences were looking at the animal models, understanding them, looking at new drugs, what do they do to animals, looking at new drugs and what they do to cell lines. And ultimately, the goal is that we end up with a treatment that we can take to patients. And so the first couple of years are those kind of what do we know, what do we not know, and what's our initial signal that we're going in the right direction. And then the next couple of years would be actual IND enabling studies. You may hear that term thrown around by your basic scientists, and that's really your specific toxicology studies. What are the short-term and long-term safety effects we can learn from our animals, either mice, rats, or monkeys, typically? What does the drug do? How long does it last? Where does it go in the body? Again, learning from our animals. And then what dose do we think we may want to do? And there's some ways to calculate that by making guesses of what dose did we use that was effective, what was too much, what was too little in our animal models, and how can we translate that into a human equivalent dose. So that takes us here. So that's all of our preclinical stuff that gets us ready to go into our phase 1/2 study. I call it a phase 1/2 because traditionally in drug development, a phase 1 study is in healthy individuals who don't have a disorder. So you're just looking at toxicity in a healthy population. And your phase 2 study is looking at safety and some efficacy in your disease population. But because these are genetic medicines, they're expensive to make and they're a little more risky than your typical, let's say, blood pressure medicine. So we don't typically go into healthy volunteers. We go straight into our disease population of interest, and we call it a phase 1/2 study. This is the initial study that gets done almost always. Every once in a while, you get lucky and you might be able to register off of this trial. But most of the time, this is your initial study, and you need a second study, which we call a phase 3 study, that is a little more rigorous, typically has a control group, and is really trying to get at efficacy. So we're going to continue to look at safety always because safety is first and foremost important. But in this phase 3 study, you're able to say whether or not something is effective compared to something that is not the treatment. Often that's either a placebo or a sham, or it's a historical control, but you're comparing it to something. And then again, I mentioned we have our phase 4 studies. Those are the long-term studies after approval. One other plug here is that clinicaltrials.gov is a really great resource for you. So I encourage everyone on the call to go to clinicaltrials.gov and just poke around, see what you can find. Start to familiarize yourself with how the website is set up, how you can search for things, and then I think there's even some alerts you can set up to know what's happening with your community. PMSF is also another great resource, of course, because they are going to have their finger on the pulse when these clinical studies open. But it's nice to have some familiarity with navigating this website before you're actually faced with thinking about potentially participating in a clinical trial or not. So that's how we kind of the landscape of where we go. Now, what does the actual study end up looking like? So as a researcher, I'm thinking about these kind of different buckets. Who am I going to study? So it is a research study, right? It's an experiment. So we need to pick the population that we think is going to be the most impactful for answering our research question. Is this new drug safe and effective? And so that means that not everybody's going to be qualified to participate in a clinical research study. And these qualification factors are described as entry criteria or eligibility criteria. The other term you may hear is inclusion and exclusion criteria. And essentially, they're just meant to define the population so that you're kind of it's being more specific. So yes, maybe we're going to study PMS, but is there a particular portion of PMS that makes more sense? Children or adults or both? Are there certain genotypes that seem like they need to be studied in a different way? Are there certain risk factors that may be medical comorbidities or medical symptoms or conditions that are affiliated with PMS that we may want to exclude or include for a various reason? And so these are all the decisions that the study teams are making in order to define what is that right population to study that's balancing that risk and potential benefit for any candidate that enrolls. Then we need to think about what are we going to measure. So these are usually called outcome measures. There's a number of things that we'll do from a safety perspective. So you're going to be asked a lot of things about potential adverse events or side effects. Did you go to the hospital? If there have been any changes in your symptoms, changes in medications, things like that, as well as a bunch of labs to make sure that liver is happy, kidneys are happy, and all these different things. But from an efficacy standpoint, we also want to make sure we're measuring outcomes from different perspectives. So what do I mean by that? So when you have an individual, especially with a neurodevelopmental disability, we want to be able to look at the individual themselves. So the actual participant, how do they perform on a given measure? That could be things like a 6-minute or 2-minute walk test. That could be a gait analysis. It could also be things like IQ testing or developmental testing and language ability testing, all of these different things where a clinician or psychologist or a rater are actually working with that individual participant to see how do they do. But we also want to get the perspective of you as the caregiver. How are things looking at home, at school? What's the feedback? How are you seeing this individual participate in their daily activities? And then from the clinician, how does the clinician, the neurologist or pediatrician or medical doctor who's following them through the clinical trial, see them change over time? And so we try to triangulate these different symptoms, getting perspectives from all these different people. The other thing you want to include, if you can, is a surrogate measure. So this could be a biomarker like an EEG. I know your group did a lot of work with evoked potentials. A gait analysis that has a quantitative aspect to it, analyzing the gait, could also be a biomarker. But the other way we think about biomarkers are blood tests or measuring protein levels in different body fluids. Admittedly, there's not that many fluid biomarkers in neurodevelopmental disabilities, but I know there's a lot of work going into it. But these are things that could be an objective measure that could then also be linked back to a clinical measure. So you see a change on an EEG, and that corresponds to a change on a developmental test. That would be ideal. So if you have anything like that, usually researchers are trying to include those in a clinical study plan as well. And then you need to think about how long do we need to follow participants. So understanding how they do over time with typical standard of care is really important. So all of that natural history work, registries that you've contributed to, medical history data you've contributed, that all goes into us understanding what does life look like with current standard of care. And then we can make some decisions. Do we need to follow patients for a couple of weeks, a couple of months, or a couple of years potentially to see a difference? And that's going to depend on the disorder and what you're measuring. And then what's your comparison group? As I mentioned in those early phase studies, phase 1, 2, you don't typically have a comparison group. It's your initial learning study. We give this treatment to participants, and what do we see change? And then we use that information to help inform our more rigorous phase 3 study, where we actually do want some sort of a comparison group. In some cases, that's an active group. In other cases, it could be a historical group. So in an active group, you're going to have a placebo or an inactive treatment like a sham that is given to one group of individuals while the other group is given the active treatment, and you compare how do they do over time. That's probably the strongest and most widely accepted by regulators. Sometimes there is strong enough historical natural history data to say this is how patients look with current standard of care, and we're going to look at our treatment group to see how they compare to that historical control. There's a lot of limitations and a lot of considerations that need to go into that design, but it is an option out there, and we're always thinking about it, especially in rare disease when our populations are smaller. So giving you a different schematic of those design options that building off of that, the placebo-controlled double-blind randomized clinical trial is really your gold standard. So you take a group of subjects in that yellow cloud, you randomize them to randomly assign them to go either to an experimental arm where they're going to receive active treatment, or to a control arm in the bottom lighter blue where they're not going to get an active treatment. In some cases, it's a placebo that's administered in the exact same way with the exact same, looking substance as the treatment or active treatment. Follow them over time and then measure the outcomes at the end or along the way and see what's changing. And the hope or the hypothesis is that your treatment arm will have benefits that your control arm does not. And then that's pretty compelling that there really is a difference, and maybe it's the treatment that's causing that difference. Now, if it's not a placebo, it could be a sham. So that's where a fake procedure is completed, but no drug is administered. There's also an option to make this an open label where they're just getting standard of care, but the treatment arm is getting treatment and the control arm is not getting treatment, and you're just observing how they do with current standard of care. It's a number of different ways to do this, but they can each have different pros and cons and levels of burden that come with them. Another way that's kind of interesting, especially to think about in gene therapy, where you're only giving a one-time treatment, is to do this randomized placebo phase clinical trial. So again, you take a group of subjects or candidates, you randomly assign them to active treatment initially or to a shorter placebo window, and then they'll end up on active treatment eventually. And so in this paradigm or in this design, everybody gets treatment eventually, but we have a randomized time point at which they're getting treatment. And so you can randomly assign this, and maybe you're able to keep it blinded so that you don't know when your treatment is happening. But when we go back and look at the data, you can start to see an inflection point where the scores are starting to change on whatever your outcome measure is at the time that the treatment is initiated. So it's kind of a staggered enrollment to treatment versus a group that just gets treated right out the gate. So these are just two options. There's a number of different other options too, and people are getting creative all the time. Just like we're getting creative with our new therapies, there's creativity in how we design these studies. But all of this ultimately gets discussed with regulators, and one of the key elements in these plans is going to be your outcome measures. Outcome measures are notoriously tricky and complex in neurodevelopmental disorders, especially because, as I mentioned, we don't typically have a lot of fluid biomarkers that we can hang our hats on to say, yep, see that enzyme levels going up and the substrate levels going down, and that's exactly what we want to see happen. It's a lot more complex in this when we are thinking about brain connectivity and learning and memory and different pathways in the brain, it's harder to nail down some of those more objective markers. But all the work that you've done on your registry studies and your natural history studies is really feeding into this plan. So you have a robust amount of data to look at and say, well, this measure didn't work, it didn't move much, or it didn't capture the right symptoms that we thought it was going to capture, but this measure did. And so all of that starts to feed into which measures you're going to carry forward with you into your clinical trial. And this is what we can think of as clinical trial readiness from the clinical aspect, kind of bookending it with our preclinical. And all of this justification and data goes into your IND to support what you think you're going to do for your clinical trials. And all of your animal data goes in to support what you're doing with the clinical trials from the preclinical perspective and justifying your dose. This is all justifying your outcomes. It all feeds together like a giant complicated puzzle piece, and is so critically important. And be proud of yourselves, because you guys have done decades worth of work that is going to be really helpful as you're entering this really critical era of new clinical trials coming around. So how do we pick these outcome measures? What do we mean by them? Biomarkers, I've mentioned a couple of times, can be anything like blood tests, electrical measures like EEG or evoked potentials, or imaging. So can you do different types of imaging than just clinical imaging? There's functional MRIs, there's retinal imaging, there's structural imaging where we can actually look at volume. You can even look at tracts in different ways, or measure substrates. And then there's functional outcomes, which are these more clinical outcomes. So you want these to help you kind of objectively say, there's no bias in this. A blood test does what a blood test does, regardless of whether we think we're on treatment or not. And a clinical test gives that blood test meaning potentially. So if we see that quality of life scores are going up and our blood test is going in the direction we want it to, then you're like, oh, then I think this treatment really might be doing something. And so they all kind of work together, and you can't necessarily have one without the other. But I will say there's a lot of times that we do not have good biomarkers in neurodevelopmental disabilities. And then when we're thinking about our clinical outcome measures, there's the functional measures. These are the things that I think about, like performance measures. So how is a patient or a participant in a trial changing in their cognitive scores, their communication scores, their motor abilities? And then the proxy report measures, because a lot of times our kids with NDDs can't speak for themselves. So as the caregiver, you're speaking on their behalf to say what's going on in their life. Does their life look better now than it did before? Does it look worse than it did before? And then the physician can also provide some input in kind of standardized scales of severity. Sometimes these are specific to the disease, sometimes they're specific to a symptom, and it just depends on the needs of the community and what measures are available and what makes sense. Also, what you've tested in your natural history studies. So a number of challenges that I've highlighted. We are running tight on time, so I'm going to go through this quickly. This takes a long time. It costs a lot of money. One size does not fit all. It really depends on the disease itself and what your treatment is going to do, depending on all these decisions that you make. And you want to get as close to Goldilocks as possible, the right gene, the right tissue at the right level for the right amount of time. And so there's a lot of complexities, and it takes a village of experts to get these things across the finish line. So quickly, what is it like to be a participant? These are experiments, they're not clinical care. So it's always so important to remember that there's a protocol, and the studies teams have to follow that protocol to the 9th degree. They have to follow it, crossing every T, dotting every I, and it may not align or be in agreement with what your clinical care would be. Your child will always be taken care of, and their safety will always be paramount, but there may be things that you have to do within the context of a clinical trial that you wouldn't be doing, and you may have less flexibility in some of the decisions you're making when you're a participant in a clinical trial. The other thing is, it's not clinical care in that we want to test an experimental therapy on the fewest number of individuals necessary in order to answer our research question. So remember, access to a clinical trial is not the same as access to a new medicine. And so while we really, as a sponsor, wearing my sponsor hat, I really appreciate all the enthusiasm to get into a clinical trial, we do have to remember that at the end of the day, they are experiments, and they're very important experiments, and we don't want to do an experiment on more people than is absolutely necessary. So again, those eligibility criteria, we're going to define the characteristics of the study population, and every participant has to meet those predetermined inclusion-exclusion criteria before they can enroll. Medical records will typically be reviewed. There's typically a lot of testing that gets done during the screening period. So even before you're actually enrolled in the clinical trial, you're going to be doing a lot of activities, and they may be asking you to dredge up old records and call old doctors to verify things in your medical history. And we typically enroll candidates who are relatively stable because you don't want to expose an individual to undue risk. So you want them to be at their best, kind of healthy relatively, unless we're over enriching for a certain symptom. But in general, they're relatively medically stable and don't have a lot of medication changes because we really want to understand what happens when we introduce this one new treatment. So the sickest, most severe candidates may have a higher risk of harm and may not be the best candidates for a clinical trial. And one of the most important things is that you, as the family, are committed to the trial schedule and are willing to complete all of the procedures that are involved in the protocol. So, no missed appointments, you're coming in early. That may mean the day before, especially if you have to fly, and that you're easily reached by phone and email to engage with the study team because they're going to have extra questions for you. It just inevitably happens. So, being as available as possible is also really important. And the schedule affects the whole family. Apologies for the typo, but it affects the whole family. So we need the same caregiver to come to every appointment. If that's mom, that means that mom and the candidate or the participant are being taken out of the house, taken out of school, taken out of work every so often for all of those study visits. And that can be stressful for the folks that are left behind in the home, too. It's additional costs. So typically, clinical trials do cover travel, lodging, those kinds of things, but and there may be a daily stipend, but inevitably you're going to be out some money most likely when you're a participant in a clinical trial. There's lost wages from lost missed time at work, using your vacation days for this. Potentially depending on your work structure. And then there's always additional costs that are kind of unforeseen whenever you're traveling. And importantly, the schedule is determined by the protocol, not by the patient or the participant or the family. So you may be asked to come in at weird times or times that aren't as convenient for you, but that's part of what you're signing up for a clinical trial. So important to go in with all these questions and really understand what your responsibilities are as a candidate and the potential participant in a clinical trial. And then to run through a few of the risks, and then we'll be wrapped up. There's no guarantees. So these are experimental treatments. Again, they're not approved therapies. And so we don't know if they're going to work or not. And so that's why we do the clinical trials. So it's important to remember that prior to approval, there's no guarantees that it's safe or effective. And even after approval, there's things that we didn't know because we didn't have enough time to know, or we didn't have enough kids to know about. And so we'll continue to learn things, and things will be updated even after approvals. There will be some degree of procedural risk because these are very rarely any oral medications. They're almost always injected in some form or fashion, and the type of procedure will dictate what those individual risks are. There's an inflammatory component. So in a lot of these, they're getting packaged into a virus. A virus's job is to stimulate our immune system. That means that we end up with an inflammatory condition or an inflammatory response to these therapies very often. That can look like fever, malaise, flu-like symptoms. But it can also mean that it triggers our immune system to recognize this therapy after it's been delivered. For individuals who have never made that protein before or have a null mutation, you could have a reaction to the body seeing that brand new protein out of nowhere. Less common in NDDs, but it can happen. And then you can also have a reaction to the viral capsid or the package. And so then it makes it, almost impossible with current today's standards to be able to administer these treatments again because the body will immediately recognize it if you give it again. There's long-term risks that are really largely unknown, to be completely honest with you. These are early days for gene therapy, and so the follow-up is typically required anywhere from 5 to 15 years. Excuse me. And so the long-term benefits and long-term side effects, we're really learning alongside the community right now. We have some guesses from our animal studies that suggest that the long-term cancer risks are variable with different types of vectors depending on what's being used. But really, it's early days and we still don't know what we don't know yet. And finally, that one-shot aspect. So there's a large immune response to that initial dose. And so we really need to get that dose right the first time. And again, we want to be Goldilocks. We don't want to give too much and cause toxicity, but we don't want to give too little and not have as much benefit as we could have had. So it's really important, those preclinical studies, that we have a really good understanding of what dose we think we're going to end up needing. And finally, what do you need to consider if you might be a participant in a clinical trial? Can you handle traveling for the next, every couple of weeks or every couple of months for the next year? What if it's the next 5 years? What if it's the next 10 years? Thinking about that and asking those questions when you're in those conversations with the study teams is super important. Study visits are long and they're stressful. Can you handle that? What if there's adverse events in your child? What if there's adverse events in a different child? What if a child dies? We know that in the Rett syndrome gene therapy in one of the clinical trials, there was a patient death, and we never want that. That's the worst case scenario, but it can happen. And so understanding all of these different risks and really grappling with it and talking with your family and your support network about where does your comfort level with different risks sit, and understanding, when would you be comfortable being a potential participant. And so the last final words I'll leave you with are that, again, if I can leave you with nothing else, clinical trials are science experiments. They're not treatment, they're not therapy. They are to test if a new therapy is safe and effective. Eligibility does not guarantee a spot in the trial, and we want to test on the fewest number of participants possible. Participating is a big decision. You really need to think about it, ask lots of questions. It's important to understand all the risks and what your responsibilities are before you commit. And then don't post anything. These are rare diseases. There's not that many patients in a lot of these studies. And so making sure that we're not accidentally exposing information before it's meant to be exposed, or potentially biasing the study and giving false hope or different hopes, because everybody responds a little bit differently. And then just be patient with the study teams, with your advocacy group, and with all the sponsors involved. Everybody's working towards the same common goal typically. And so, just be as patient as possible. It's a long, arduous process, and we really appreciate your participation. So with that, I am going to stop share. And I went way over time. I'm so sorry. We have 10 minutes for Q&A. We were supposed to have a little bit more, so I apologize.
That is okay. Thank you so much, Dr. Goodspeed. That was a fantastic presentation and really thorough. So I hope all of our attendees were able to enjoy that. We do have a couple questions coming in. Hopefully we can keep it to 10 minutes. If people are okay going 1 or 2 minutes over, we'll stick around just to make sure. And remember to use that Q&A function if you have any additional questions. And if we totally run over, we will make sure things get answered at a later date and publish a document or something.
Good.
So one of our first questions that I know has been on a lot of our families' minds is that oftentimes clinical trials ask for younger children as participants. So does this mean that there's less of a chance that for instance, gene therapy might work in an older individual, or why do some trials start in younger versus older participants?
Yeah, it's a great question. And it's been kind of an evolving issue. So when I first started in gene therapy, about a decade ago at this point, which is crazy to think about, we were getting a lot of feedback from FDA that if there were adult individuals with a disease, you needed to go into them first to look at safety. But I think that there has been a bit of a shift that in medicine, with almost anything, right? Even if you think of the common cold, if you catch it early, you may have a better response to whatever that treatment is. And so there's this assumption in gene therapy that that also holds true. I think it's still early days and we don't know, especially when you've got neurodevelopmental diseases. This is my personal opinion, but I think when you've got a neurodevelopmental condition and you don't have degeneration where cells are dying, so neurons are dying, for example, in a metabolic or degenerative condition, then you have a bigger window of opportunity to treat. And so my bias has always been, if we don't have evidence of cell death, then you can't tell me that an adult couldn't benefit. Um, would a younger child benefit more? Possibly, but I don't think we know yet. And so I think it's just dependent upon the sponsor, and it's also dependent upon what that preclinical data looked like. Did they look at younger animals and older animals? Did they only look at younger ones? So it's good questions to ask whenever a new treatment is coming up and you're seeing that the inclusion criteria have been pretty restricted to younger kids.
Thank you for that answer. Very thorough. Another question that I really like that kind of came in is, there have been gene therapies that have been, trialed in other rare disorders like Angelman syndrome. And so what can we in the PMS community sort of take away from that? And is there anything that we can sort of apply for our current and future trials?
Yeah, so I think when we're thinking about neurodevelopmental trials, it's good to think about regulatory precedents. So regulators like to apply the same logic to different disorders over time. And so when they have something that works, they like to apply that again and again and again. So right now, we've got a situation where the very first drug was approved for a neurodevelopmental condition for Rett syndrome. That was Daybue or trofinetide, small molecule, oral medication, not a gene therapy. But what we can learn from that is, what did the regulators see in the data that they thought was compelling? And then can you apply any of those principles to studies that we're doing in other neurodevelopmental conditions? Right now in Angelman, we have a nice agreement to use the Bailey scales of infant development, as well as this kind of responder analysis that we do at Ultragenyx. It's a really cool tool called the MDRI, or the Multi-Domain Responder Index. And we've got nice agreement to use those two tools to test efficacy. And so I think those are the main things that you can learn is, how are they designing their trials and what endpoints are they picking? And is there anything that you can start to overlap or point to those as test cases to say, well, so-and-so did it. So like in Angelman or like in Rett, we are using X, Y, and Z outcome measure.
Great. We have a couple additional regulatory-based questions. So one of them is, for example, once a drug, let's say, is approved in this country, what does it take to get approved in another country? Do you start from zero, or does that sort of speed up the process? If you know.
It depends on the country. So there are some countries where, or regions, because for example, Europe has its own, like the EMA, the European Medical Association. The UK is separate, so they have MHRA. Then there's Health Canada, there's Australia, Japan, so and so. So each region will have its own little nuances of what they expect, what they want to see in the data, and it's really going to depend. So that is the wishy-washiest of answers, but the real answer is, it totally depends. It depends on the risk profile of the drug. It depends on the risk tolerance of the country. And there are certain countries that do require evidence of data in their population before they will even entertain an application.
That's helpful to know. This one seems a little bit harder to answer. Someone was asking, how much could gene therapy cost once it's approved?
I think so. I learned back as a little med student, when you're faced with hard questions like this, you can frame things. So we're talking days, not weeks, or things like that. We're talking millions, not hundreds. Probably not even thousands. We're talking millions. So the price tag is really high right now, and it's the manufacturing costs mostly that are driving a lot of that cost. But it's also, every step of the way takes a lot of money to get these across the finish line. So because it's early days, you may have to do extra toxicology studies compared to a hypertensive drug that's been around, and maybe there's some precedent that you don't need as many studies, preclinical studies, to go into humans for that. But because these are all new, There's a lot of additional preclinical studies. Monkey studies are really expensive, but they're really important and informative. Clinical trials are really expensive, but really important and informative. And then the manufacturing costs are still quite high. Probably going to come down, but they're still quite high. And so all of those things add up to the overall cost of a drug. And then there's a lot of arguments about, well, if we can change the trajectory of a disease, do you offset costs in other ways because their symptoms are less later on? Those are all some of the arguments that folks are making. I encourage everybody to attend ASGCT because there's almost always a session on this, and it's always fascinating to hear it from a lot of different perspectives.
Yeah. Let's see if we can do maybe two more questions. Some questions in around sort of the operation for gene therapy, and sort of what that looks like. Is it always, in the brain or the spinal fluid? How long is like a stay expected?
So it'll depend on the different modality. So for us, for our ASO, I can speak to that because that's what we're working on right now. It's a procedure that takes about 15-20 minutes to infuse. You get monitored for a couple of hours before and after, and kids go home the same day. For a gene therapy, typically you've got an overnight stay around the time of the infusion. And that's because they can typically have a really big inflammatory response. And you want them to be in a safe environment where they can manage any of those symptoms that come up. So typically a gene therapy program, you've got some pre-medications you're going to be taking for days to weeks to coming before the infusion. The infusion itself is short, but you're probably going to stay in the hospital at least overnight, maybe for 2 or 3 nights, kind of depending on the protocol. And then for gene therapy studies, they often want you to stay local to the site for weeks to months after the infusion. And that's again for that close follow up in that critical window when we know that the inflammatory response can be the highest. So you're on immunosuppressant drugs typically. And you are also having frequent follow-ups for blood testing and check-ins with the physician and study team to make sure they're safe. So it's important things to consider as you're looking at a potential protocol or a study to join, that you're understanding what are my commitments. So for our ASO, it's a day procedure, but you're coming monthly at first, and then it kind of lessens. For a gene therapy, it's a one-time infusion, but that one-time infusion may come with a couple of nights in the hospital and a couple of weeks to months on site, or local to the site.
Great. And so one of our final questions is for this, for example, if you have two mutations on the same chromosome, could you kind of use the same gene therapy at the same time if it was kind of targeting both of those? Basically, can several gene therapies kind of be used either at the same time or different times if there's multiple mutations?
There was a lot of really cool talks about this at ASGCT this year. So it's really incredible what the basic scientists are able to come up with and all the engineering they're doing in the lab to address these really complex genetic disorders. So I will leave it at that I think the sky's the limit for what scientists are going to be able to come up with to do. Right now, most of the therapies that are going into clinical trial are really targeting a single gene in a single way. But I do think there's some really exciting stuff coming up in the future and on the horizon. And we don't know what we don't know yet, right? And that can be good or bad. So stay tuned. I think there's a lot of exciting stuff coming up the pike.
Wonderful. Well, thank you so much again, Dr. Goodspeed. This was an incredible presentation, and I hope our families learned a lot, heard enough of knowledge that they were maybe previously new, and got some new knowledge out of it. So we're really grateful, and a recording of this will be available for our families afterwards. And again, also thank you for AGS. Oh my gosh, I am totally going to butcher that. Allie, thank you as well for being here. And feel free to say any last minute words. We really appreciate you.
Oh, thank you guys so much. This was so much fun. Thank you to the community for listening, and for having such awesome questions. Thank you. Huge shout out to Allie. She makes so much magic happen behind the scenes. No, it was a pleasure. Thanks for the opportunity.
Absolutely. Thanks all.